Hello, everybody. Welcome to our next general biology lecture. Today we're going to be switching gears from talking about the flow of energy and matter through the system and cellular respiration and photosynthesis. We're going to be talking more about the flow of information through biological systems, what is referred to sometimes as the central dogma of molecular biology, how we have genetic information getting copied, replicated, and translated into proteins. So today we're going to recap some about DNA and how it's structured, and get into talking about how it's replicated. This is our general overview of our topics for this lecture. Please use this to guide yourself to jump ahead to different parts. But let's start off with our recap of what we learned in our macromolecule lecture. So remember that nucleic acids are polymers that are used for data storage and transfer. They're polymers composed of monomers called nucleotides, and each nucleotide is a nitrogenous base and a pentose sugar and a phosphate group. So that 5-carbon pentose sugar is either a ribose or a deoxyribose. Then it has this nitrogenous base that is attached to that first carbon on the pentose sugar. We usually, in this view of that sugar, we start counting the carbons next to the oxygen. You can remember that the 1 is where the nitrogenous base is attached. Then you go 2, 3 prime carbon, 4, and then 5. The 5 prime carbon is the one that's kind of sticking outside of the sugar. So the ring of the sugar, one of those parts of the ring, is the oxygen. And then the phosphate group is attached to that 5th sugar that's sort of sticking out. 5 prime, 3 prime, those we want to keep track of going on. So when they're combined, they form polynucleotides, many nucleotides together, which take 2 different forms. We have deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. Also recall from our last few weeks, we've talked a lot about ATP and NADPH and these other coenzymes, these other cofactors that carry energy. Those are nucleotides, and they're actually precursors to the building of nucleic acids. But those nucleotides have more phosphates, and they have different structures that make it to where they don't form polymers. They don't form nucleic acids. But important to note that they are similar in structure. A little bit more on the structure of the nucleotides. So again, DNA and RNA, they're named for the type of pentose sugar that's in the middle of them. Ribose has an extra hydroxyl group there, whereas deoxyribose has deoxy, is missing that oxygen. And then that nucleic acid is really named for, or the nucleotide is named for the type of nitrogenous base it has. So that shouldn't say nucleic acid, that should say nucleotide. Actually, I'll correct that later. And these nitrogenous bases, they themselves have a few different forms. There are the purines, which are the 2 rings, these carbon-nitrogen rings. So adenine and guanine, they're actually at the bottom here. So they have the 2 sort of attached rings to one another, sort of like a 5-ring and a 6-ring, pentagon and a hexagon. So you have cytosine. And then you have your pyrimidines, which are cytosine, thymine, and uracil. Slight differences in their structure. Cytosine and thymine are found in DNA, whereas cytosine and uracil are found in RNA. So uracil and thymine, uracil takes the place of thymine when we're talking about RNA versus DNA. They were mainly talking about DNA, though. Mostly. So yeah, when we're looking at the overall structure of a DNA molecule, it is structured in the form of a double helix, these 2 strands of DNA that are winding around one another, with the phosphates and the sugars forming that backbone along the outside edge. And then on the inside are the nitrogenous bases that are paired up with one another in a complementary way, depending on their hydrogen bonds. So they're forming hydrogen bonds with one another, which remember aren't really bonds per se, like they're not wholesale sharing electrons, but they're having an interaction, an electrostatic interaction with one another. But they don't do this in a random way. So adenine always pairs with thymine, cytosine with guanine. Of course, adenine does pair with uracil when we're talking RNA, but what it comes down to are two things. So it's the structure, the size of the molecule. So we only have a purine paired with a pyrimidine in both these cases, and then you have the purines. The ones that form two hydrogen bonds are capable of forming two hydrogen bonds because they have these nitrogens, the way they're paired up. And then guanine and cytosine are able to form three bonds. So guanine's our purine, our larger one with the three bonds, whereas adenine is our larger one with two bonds, cytosine smaller with the three bonds, thymine smaller with the two bonds. Same goes with uracil there. The other part to note here, again, is there's a direction to this where it kind of goes in an opposite direction where that 5' end is going up in the upper left in this case, on the left strand, and on the strand on the right, the 5' is on bottom right, and then the 3' is up there. So the 5' again is where the phosphate is. That phosphate will then connect over to another sugar, another pentose sugar, and that is how these nucleotides are built together. And we'll talk more about that. But note that there is a directionality, and the strands run in opposite directions, sort of anti-parallel. Quick note on RNA. We'll talk about these more, especially when we get into talk about transcription and translation, but there's 4 different types of RNA. We have our messenger RNA, which carries information from DNA. So it's involved in carrying that information over to the ribosomes, where it will be translated into proteins, into amino acids, which get formed in proteins. The ribosomal RNA is a key part of the ribosome itself. It's what enables the messenger RNA to interact with the other parts of the ribosome to form the overall interaction that attaches the amino acids. Transfer RNA brings those amino acids over to the ribosome to where they can be used to synthesize the proteins. And then there's microRNA, which is involved in regulating which genes are expressed and like influencing the rate at which different what am I saying? The rate at which different strands of DNA are transcribed and translated. So 4 different types of RNA. We'll talk about them more later. Oh, but important to note here, remember RNA is only a single strand, so there is no double helix. It usually just forms these single helixes, and then the nitrogenous bases are interacting with other RNA, like sort of on a case-by-case basis, but they don't form this stable dual helix molecule structure that DNA has. So, recap, these should be pretty similar notes to what you have from our macromolecules lab, but remember that they're what DNA is, what the different types of RNA are, the difference between the 2 purines and the 3 pyrimidines, and then what are complementary, which ones are complementary with one another. A to T or U, and C to G, always. G to C, T to A. So let's talk about the overall genomes and how they're structured, the organization of all the genes that organisms and cells have. So that complete set of all the different types of DNA that an organism or a cell has is what we call the genome. This is divided into individual single DNA packages in that overall set. That's what we call a chromosome. Each chromosome is just one single long DNA molecule. But then got to keep track of numbers, whether it's like what's been duplicated and like the different parts of it, but we'll get to that. When it comes to prokaryotes, they're simpler and more complex in ways. Simpler in that the chromosomal DNA that they have is usually, most prokaryotes have a single loop of DNA, a single chromosome that is actually like a circle. It's located within a nucleoid region because they don't really have a nucleus, they don't have membrane-bound organelles. So its nucleoid just means like a nucleus. So they have a little area of the cell that they keep their DNA in. But then where prokaryotes are more complicated is they often have these secondary loops of DNA that are outside of the chromosome that are called plasmids. These are hugely important to how bacteria exchange genes among one another. Bacteria and archaea, all prokaryotes, in this process called horizontal gene transfer. It's also key to a lot of our biotechnology methods when we're looking to insert DNA to replicate DNA with the help of microbes. So, important to keep in mind that these plasmids are these little extra loops of DNA that prokaryotes have. And then you have us eukaryotes, us plants, animals, fungi, etc. We have much larger genomes than prokaryotes, and that most of those genes are located within the nucleus of the cell as a membrane-bound organelle. We're eukaryotes. We do have some DNA that is in the mitochondria as well. These small circular chromosomes that have genes that help code for the proteins involved in the electron transport chain and oxidative phosphorylation. The origin of those probably has something to do with the origin of the mitochondria itself as an organelle, the origin of eukaryotes as incorporating prokaryotes into larger cells as organelles. Hence why they have maybe similar circular structures. But we have a lot more DNA. In order to store up all that DNA, it ends up getting wrapped around proteins called histones, forming these individual nucleosomes, that those end up interacting with one another, as we'll see in a video a little bit later, to form a substance called chromatin. That gets wound up, more tightly packed. And what this means is the winding up makes it less accessible to be replicated or translated, but it also makes it less vulnerable to damage when these chromosomes need to get moved from one place to another, such as in the case of cellular replication or division. So more stored up is ready to be shipped. And when it's in those more stored up bundles, we refer to these chromatids, these sticks, these arms, each of which are called a telomere, that are attached to the center by a centromere. So these different regions of the DNA. This is all still one chromatid, is all still one big DNA molecule that is just bundled up very tightly with the help of all these proteins. Think of those histones, those proteins, as like sort of wrapping thread around a spool in order to make it more tightly compact and organized. And then those centromeres, these different regions end up being involved during cell division. And during division, each of these chromatids actually gets duplicated into this sort of, looks more of like an X shape in most of our human chromosomes. So when you see that X, that's a chromatid, a unduplicated chromosome that has been duplicated to have both two sets of the same DNA. So each chromatid is one DNA molecule, one unduplicated chromosome, but then that can get duplicated, and then we use chromatid to refer to half of that duplicated chromosome. But then we have each chromosome we have multiple sets of each of these. So a diploid cell is referring to a cell that has two matching sets, diploid, di being two. So two matching sets of homologous but unduplicated chromosomes. So each of these chromosomes are homologous in that they're similar length and contain the similar sets of genes, but they're not identical. They're also not duplicated. The duplication only happens when we're getting prepped to divide the cell. So for a diploid cell, you have two sort of similar sets of the same chromosomes. But then a haploid set of chromosomes in a haploid cell contains those chromosomes that don't have their homologous, similar structured, similar function chromosomes. They only have half of that. So this is just half of the full set. So what that looks like with our human chromosomes. So this is the genome of a human, how it's generally structured. All these sorts of things have variety to them. This isn't 100% of the time, obviously, but that's the case in everything that we're talking about in biology. There's always exceptions to the rules. But when you lay out the full set of chromosomes in a cell, that's what you call a karyotype. It's just this sort of this displaying of each of those items in the set. So we have 23 different homologous pairs in the diploid cells of a human. We have 23 pairs of unduplicated chromosomes. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, all the way up to 22. And then the 23rd, these are homologous in ways, but we tend to have, these are our sex chromosomes, our X and Y. And yeah, with all of these, typically there's just a pair of two, but there you can have 3 chromosomes in these various positions, but we organize it into the pairs. These are all pairs of homologous chromosomes. But then in our haploid cells, you don't have all those pairs. So you only have, in this case, I think it's just showing the one on the left in all of these. But we'll talk about how those get split up during cell division and meiosis in particular. So full set and half set. Remember, these are all still, each of these chromosomes is unduplicated. Something to note. So diploid chromosomes are found, you have a diploid set of chromosomes when we're talking about as eukaryotes. In most of our body cells, just the somatic cells, most of the cells in our bodies, I say our when we're talking about plants and animals, most of our body cells are diploid. So we have 2 full sets of homologous chromosomes. We have the matching pairs. But then in our gametic cells, our sex cells, our sperm, and our eggs, for most plants and animals, those are going to be haploid cells. They only have half the amount of chromosomes. But then it's interesting to note how there are whole other organisms, including some plants and a lot of fungi and things like jellyfish, where they go through a whole stage of their life where most of their body is made up of haploid cells, where they don't have both the sets of chromosomes from both parents. They don't have diploid chromosomes. And then there are whole other organisms, especially a lot of plants. You can get tetraploid. You can get 3, like you're not having a pair in this case, you're having sets of 3, sets of 4, sets of 5, sets of 6. You can have triploid, tetraploid, in a variety of our conditions as humans. Even we also do have triploid chromosomes in certain cases. So yeah, diploid, haploid. Haploid, we say haploid because it's half, but in most cases, this is monoploid in our case. But if you have a tetraploid organism, that its haploid cells would have two sets. So talking a little bit more about us humans, again, we have 23 different sets of chromosomes. Typically, these are diploid chromosomes in most of our body cells. So given that we have 46 chromosomes total, how many, just thinking about the numbers here, total, add them all up, our gametic cells would have our haploid cells, so they would have half the total number of chromosomes in our diploid cells because we are diploid organisms. Our gametic cells have half of that. So each of our gametes will have only 23 chromosomes. But if we have 46 chromosomes, how many homologous pairs of chromosomes do we have? Well, that would be the same kind of thing. We have 23 pairs, which comes out to 46 total. We are diploid. Our genes all are paired up. If we were triploid organisms, then we would have a third of them, a third would be our sets. Anyway, so we want, at this point, checking in, we should have some notes about what defines a genome. Remember, it's that full set of genes that an organism or its cells have. We have chromosomes and chromatids. A chromatid is essentially half of a duplicated chromosome. So that's to say, a chromatid is a chromosome itself that has been attached to one of its copies. When we see that duplicated X chromosome. And then remember the difference between diploid and haploid. Then there are some other terms that we need to clarify the meaning of before we keep going. So a gene itself is just a region of a chromosome that provides the code for a particular physiological expression. You can think of these as an individual word or phrase of information that will code for particular proteins that will lead to some sort of physiological expression. So then we talk about where the locus of a gene is as just that location of the gene on the chromosome, its position on the chromosome, which remember, a chromosome is just a long, tightly wound or variably wound up DNA molecule. So our locus is that position along that strand. And then an allele is, we have different versions of those genes. So it may be coding for a similar sort of expression, but just a different variation of the proteins, variation of the overall outcome, and different functions, slightly different functions of that same overall gene. So variance on the word, so to speak. So a gene is consisted of simply a pattern of nucleotides. So different changes in those patterns will result in different alleles, different amino acids that get translated from these genes. And those slight changes, those are what we call mutations. And we'll talk about those more, especially as we get into talking about transcription and translation. But a gene is just a pattern of nucleotides. It's a string of letters put together into a phrase. So when we're presented with these, we want to think back to the complementarity of those nucleotide bases. When we have a single strand of DNA, such as ATC, GGATA, we want to be able to think about what the pairs of this are, so A goes to T, T goes to A, C goes to G, G goes to C, and so on. So for this first one, we should get something like T, A, G, C, C, T, A, T. So work through the rest of these on your own. Feel free to check your answers with me later. So another quick check. We should, at this point, have some notes on what defines a gene, an allele, and a locus, or loci, plural. So remember, genes are the full just patterns of individual nucleotides strung together into a word or a phrase. As an analogy, an allele is just a variation of a gene, a different version of a gene, because not all genes can be similar without being identical. And then they can be located at the same locus, the same place, but just have that variation to them. So we'll finish up here on talking a little bit more about that overall organization of the chromosomes, how they're replicated. We'll work through some of this in our lab as well, and we'll watch a couple of these videos real quick here. But when a chromosome is not being moved around, when it's being used, those chromosomes have to be unraveled. This is what exposes the nucleotides and allows those genes to be expressed, to be transcribed and translated, that flow of information. But then when those genes aren't actively being expressed, when the chromosomes aren't in use, they're stored by being wrapped up. Again, talk about those histones and the chromatin. And then this is what's necessary in order to get it into a much more compact, shape that can be more easily transported without getting damaged. So let's watch this short video that will talk about this process.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
So it's not quite true to say that the chromosomes don't exist when a cell is not dividing, but they only exist in that really highly structured, highly compacted state and duplicated state when a cell is getting ready to divide and is actively dividing. When it's done, it starts to unravel so that the new cells can be more those genes can be expressed in the new cells. And one of the key parts in getting ready to divide and building up before the chromosomes can be divided up into new cells is you got to make a copy of it. So we're going to be talking about how to the process of DNA replication, by which you go from an unduplicated chromosome to a duplicated chromosome. There are errors that happen along this process that lead cause mutations in this replication, but this process is pretty accurate because, as we'll talk about, DNA polymerase is very good at its job and has its own built-in proofreading ability. So just watch this last video here.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand. And wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called kinase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in a 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
So yeah, there you have it. That is the process that is going on constantly within our bodies as our cells are dividing and replacing themselves. Because in order to divide, you first have to create a new copy of DNA to go into those new cells. And of course, yeah, there are errors that happen sometimes that cause mutations and like the misforming of proteins or the non-forming of proteins. And we'll be going over this in the lab section. There's a video we watched in the lab section that covers this a little bit more in a different format. But there you have it. That is the process of DNA replication that is going on across millions of base pairs within each of our chromosomes in preparation for cellular division. So we want to keep track of those little steps, note how there are errors that can occur in each of those, all the different players involved. There's all these different enzymes. There's a few different enzymes in particular that we want to pay attention to. DNA helicase, which does the unzipping, DNA polymerase, that does the building. But the thing is, it can only build it up, as they said, from the 5' to the 3' end. So those nucleotides only get added on that 3' carbon by DNA polymerase. So you end up with these Okazaki fragments on one of the strands, on the lagging strand, where it had to be built up sort of piece by piece. And then you have those other enzymes that help clear things up and fill in the gaps. Big process that has to go on for millions of base pairs for each of our chromosomes. So that's all I have for you today. Hope to see you in class, and take care.